In this section, we will talk about the physiological properties of protein molecules. Protein has many roles in your body. It helps repair and build your body's tissues, allows metabolic reactions to take place, and coordinates bodily functions. In addition to providing your body with a structural framework, proteins also maintain proper pH and fluid balance. Homeostasis is essential for the normal function and formation of the protein molecules. Let's start by discussing the basic characteristics of proteins first. So we know about amino acids, and that these are the monomers that will form proteins, which are also known as polypeptides. Proteins are polymers of amino acids, and they are the most diverse type of biomolecule in your body. Different kinds of proteins include enzymes that catalyze chemical reactions, receptors that control signaling in your body, hemoglobin, which carries oxygen throughout the bloodstream, muscle, and organ tissue, which gives your body structure and mobility, and so many other things that are essential to maintain homeostasis. To understand their physiological structure, we will discuss how the polypeptides chains are formed to make a protein molecule. So how do amino acids polymerize? This happens when amino acids form peptide bonds with one another, such as the peptide bond between these two amino acids shown here. Peptide bond formation is an example of a dehydration reaction, so a water molecule is lost, and these two amino acids come together to form a peptide bond, which results in an amide. An amide is a functional group with a nitrogen atom next to a carbonyl, and this is the functional group that will connect each amino acid during polymerization. If two amino acids combine, we get a dipeptide. If between 3 and 10 come together, we would call that an oligopeptide, since oligo means just a few. And if more than 10 come together, we will call that a polypeptide, since poly means many in proteins are large polypeptides of around 300 to 1,000 amino acids that are folded in such a way that they have some biological activity. Proteins are very large compared to simple molecules. They contain hundreds of amino acids, and they have very specific shapes from which their function is derived. So let's learn about protein structure. Protein structure follows a specific hierarchy, so let's start first with primary protein structure. As we have already discussed that two amino acids come together to form peptide bonds. The primary structure of a protein is simply the sequence of amino acids with no attention paid to any aspect of three-dimensional shape. The shape the protein will take depends on the primary structure because it is the identity of all the side chains in the sequence they are found in that determines how a protein will fold up. To see this in action, let's look at secondary protein structure. Secondary protein structure describes localized conformations of the polypeptide backbone, meaning the folding pattern that a protein will exhibit over a few dozen amino acid residues. So from the primary structure, we get the secondary structure, and this comes from hydrogen bonding interactions. So here we see intramolecular hydrogen bonding. The secondary structure of protein is in the form of alpha helices and beta sheets. The alpha helix and beta pleated sheets come from hydrogen bonding between the partially negative oxygen on the carbonyl and partially positive hydrogen on the nitrogen atom. In the folding pattern, the linear amino acids transform into spiral conformation that is the helix conformation. The amino acids in alpha helix are arranged in a right-handed helical structure in which the helix has 3.6 residues. Or the linear sequence of amino acids transform into sheet-like structure called the beta sheets. You can see in this diagram, unlike the alpha helix, the beta sheets form the extensive intramolecular hydrogen bonding. Despite this structure of protein is not stable, and it needs to progress to tertiary structure in order to become functional. Now let's move on to tertiary protein structure. Tertiary protein structure involves the further folding of the polypeptide chain to produce its overall three-dimensional structure. This folding is not random. It is specific to the protein and will occur in that specific way every time that protein is formed, since it is this shape that gives the protein its function. There are a few factors that influence tertiary structure. First, residues with hydrophobic side chains, like alkyl groups, tend to be found in the interior of the protein, so that they are not in contact with aqueous solvent. At the same time, residues with hydrophilic side chains, ones with formal charges or dipoles on them, tend to be found on the surface of a protein so that they can make dipole-dipole or ion-dipole interactions with water molecules. All of this is just a way for the protein to maximize the electrostatic interactions it can make in solution, which is a big factor in determining the way the protein will fold. Another motif that stabilizes tertiary structure is something called a disulfide bond. This type of bonds is formed between the amino acid cysteine. Overall, if the protein is highly folded and compact, we would call it a globular protein, whereas if it is long and spindly, we would call it a fibrous protein. Fibrous proteins are generally composed of long and narrow strands and have a structural role you can remember them as. They are something. Globular proteins generally have a more compact and rounded shape and have functional roles you can remember them as they do something. These two are the functional forms of proteins. They have different sets of properties. You can memorize the difference between these two classes of tertiary proteins with help of a mnemonic. SPADS, or simply SPADES. The shape of the fibrous protein is narrow and long as compared to the round and spherical shape of globular proteins. Fibrous proteins are found more as structural proteins, such as those of the protein channel of cell membrane, as compared to globular proteins that mostly act as functional protein. The acid sequence, or the primary structure of fibrous proteins, is repetitive, while for globular proteins it is irregular. Fibrous proteins are more durable to changes in pH, temperature, while globular proteins are more sensitive to these changes. Examples of both these types of proteins include collagen, myosin that is actin, keratin filaments, and enzymes, hemoglobin, insulin, respectively. The fibrous proteins are generally insoluble in water, while globular are soluble. Lastly, we can examine quaternary structure. Some proteins are just one continuous polypeptide chain, but some proteins involve multiple polypeptide subunits that can come together to form a larger protein. It is the arrangement of these subunits that determines the quaternary structure of a protein, like hemoglobin, which consists of 4 totally separate polypeptides arranged in a specific way. If a protein is comprised of just one polypeptide, then it will not have quaternary structure. So to summarize what we have discussed so far, primary structure is just the sequence of amino acids. Secondary structure is the way the chain begins to fold on the localized level, forming alpha helices and beta-pleated sheets. Tertiary structure is the complete folding pattern of an entire polypeptide, and it is the functional state of a protein molecule. And quaternary structure is the way multiple polypeptide subunits come together to form a larger protein. Since we have talked about the structure of proteins, it is imperative that you should know what would happen if an alteration took place. It is important to understand that even a tiny change in primary structure can completely change the overall protein. For example, sickle cell disease is a genetic disorder where just one amino acid residue in one of the subunits of hemoglobin is changed from glutamic acid to valine. Because the side chains of those 2 amino acids are so different, the mutation changes the folding pattern at that location, and the resulting hemoglobin protein, which is responsible for carrying oxygen through the bloodstream, takes on a different shape, causing the red blood cells that contain hemoglobin to look like tiny sickles, which can then clog blood vessels. So we can already begin to see why understanding the structure and function of biomolecules is crucial if we want to understand health and disease. So that was all about homeostasis, components that make up the homeostatic system, the mechanism of actions of two feedback systems to better understand homeostasis, and physiology of proteins. Stay tuned to sqadia.com to watch more knowledge-filled videos.